Before I get into my talk, though, I'll acknowledge the funding sources that made it possible. So this was possible through a couple of NSF grants. As you'll see shortly, there's a fair bit of small angle X-ray scattering, which was primarily done at Argonne National Laboratory. And so I want to acknowledge the DOE funding there. Startup to help get this project off the ground. Funding came from Bucknell University, and then Creighton, Carrere, and Son of Orm provided materials for these studies. Lastly, I'd be remiss not to mention a couple of my co-authors here on the title slide, Lucas Rankin on the right and Will Walker on the left. These are two master's students who worked closely with me on developing the understanding that we did in this project. All right, so let me give a little context, a little historical context for thermoplastic elastomers in the polymer morphology group. This is at least what Rich's group was called when I was at NC State, and so I used that group name in the title here. The micrographs in the bottom left of this slide are from one of the first papers, as far as I could tell, that Rich published in the area of thermoplastic elastomer gels. And this is with one of his first PhD students, Jonathan Lohr. And here we see a combination of a triblock copolymer and aliphatic oil. And the end blocks of the triblock copolymer are stained so they're dark. And we'll get into that more in a minute. But my kind of collision with thermoplastic elastomer gels in the polymer morphology group began basically immediately when I arrived at NC State. I started in November of 2011 in Rich's group, and I started kind of the completion of a project that was already ongoing when I arrived at NC State, which was work with Michael Dickey on incorporating liquid metals into thermoplastic elastomer gels to make flexible electronics. And so just to give you an idea, here's what I looked like at the time. This is from a group picture. And since I was only 8 years old, I don't know what Rich looked like at the time that these micrographs were collected, but this is the picture I have in my head of Rich during this time of about 1996, when they were beginning work on thermoplastic elastomer gels. Just a quick aside here, this cropped image is from a group picture from spring of 2012. So there's some familiar faces here, maybe to some of you. I think this was a dinner celebrating Kristen's PhD defense, and so maybe this picture is meaningful to some of you. But this was early on when I joined Rich's group. And so I'm going to jump to context in another avenue, which is applications of thermoplastic elastomer gels. These materials have been used in a lot of different areas thus far. And if you're less familiar with thermoplastic elastomer gels, you might have actually interacted with them more than you think. They crop up in places like gel candles, which are not as popular as they used to be, but these are candles that kind of have a jello-like consistency, but still burn like a traditional wax candle. Consumer cushioning, such as gel insoles for sneakers or gel wrist rests for keyboards, mice, bike seats. They've also been used for ballistics gels or as surgical dummies because of their similar consistency to human muscle. And then lastly, they've also been used for hydrophobic fillers and things like transoceanic optical cables. Now, kind of in contrast to this, drug delivery media, which takes on a number of different forms, whether dispersed in solution or just plain solutions or gels, all basically make use of situations where water is in the largest abundance. And so there's a contrast, again, like I said, between the left, which is based on hydrophobic, typically aliphatic solvents, nonpolar solvents, and the right, which is heavily based on water. And so the work I've been doing the last few years with my group at Bucknell University is trying to push hydrocarbon-based thermoplastic elastomer gels into the drug delivery media space. And this image actually looks similar. It's funny, with our previous speaker who was speaking about adhesives on skin, the primary application we're talking about for these materials within drug delivery is transdermal drug delivery at this point. And so this just gives you an idea of what one of these thermoplastic elastomer gels look like stuck to skin. So this is actually my arm and one of the gels that we work with in our lab. And so why transdermal delivery? Well, that's kind of the easiest stop as far as drug delivery media go. It's on the exterior of the skin. It's not inside of the body. And actually in 2013, there was an initial paper which looked at thermoplastic elastomer gel type materials for transdermal drug delivery. And what this paper did is it combined classic kind of thermoplastic elastomer, which is styrene, isoprene styrene, shown by SIS here, along with a tackifying resin to increase adhesive performance, liquid paraffin, and their kind of probe molecule or probe drug, which was testosterone. And this study does a number of different things from a statistical standpoint. It's actually done by a pharmaceutical science department group, and so a lot of it's focused on the application of these materials. But basically the main findings from their study are that in vitro, the release of testosterone with time and the amount of testosterone permeated through, in this case, pig's ear skin, are very similar to an on-the-market testosterone delivery patch, which is called Testo patch. And then in addition to this, they actually compared, and I know there's a lot of data here, so I'll draw your eye to the squares and diamonds towards the top of this graph, an in vitro comparison between their experimental patch and this commercially available patch, which shows the plasma concentration of testosterone in mice is very similar, whether you're using their experimental patch or what's already available. And so this gives a lot of promise to these types of materials for transdermal type applications. Now, like I said, this was done, there's been a few studies like this that have been done by pharmaceutical science students and PIs primarily. And so what we're really bringing to this area is a bottom-up approach to understand the fundamental principles that dictate the performance of these transdermal patches. And so basically what we do in my lab is we combine materials that can compose a transdermal patch. And so things like a triblock copolymer or a thermoplastic elastomer, some aliphatic mineral oil, and then some probe molecule, which I'll get into more in a minute. We create a gel from those materials. We do extensive structural analysis, primarily through small angle X-ray scattering. And then we also characterize the transport properties of the probe through these materials in hopes that we can develop very extensive formulation structure transport relationships and really inform very specifically pharmaceutical groups or companies how they could design patches to achieve the goals they want, depending on what probe they're trying to deliver or the part of the body or even the various model system they're trying to deliver to. And so, like I said, we're trying to build some foundational knowledge in this area. And so here's just some basic information about thermoplastic elastomer gel nanostructure. The big things here are, this is the micrograph from before, from Rich's first work, one of his first works in thermoplastic elastomer gels. And then the schematic on the left is kind of what we imply or envision the nanostructure as looking like. Some of these features are difficult to ascertain as far as microscopy or scattering goes, and simulation really helps us to understand those things. And so our gels are composed of styrene, ethylene, butylene, styrene, triblock copolymers, as shown on the top right here, and then an aliphatic mineral oil. This is kind of a generic structure. This isn't the specific structure of the mineral oil we use, but this is kind of, again, a general capture of what we think it kind of looks like. It's primarily aliphatic, and it has a molecular weight of about 500 grams per mole with this kind of aliphatic structure. And so in the picture on the left, important things to note are the purple regions that I'm showing are styrene physical crosslinks. They're glassy at room temperature, and those map over to the dark regions in the micrograph on the right bottom side. In addition to those glassy regions, the mid-block chains of the polymer interconnect the physical crosslinks, which yield us at specific conditions, formulation conditions, a physically cross-linked gel. Now, these materials are very tunable. We can change how much polymer is in the gel. We can change some characteristics of the oil. We can change the composition of the block of polymer, so what fraction of it is end blocks versus middle block. We can also change the molecular weight of the triblock copolymer. So there's a lot of handles in terms of formulation tuning that we can do to manipulate our base gel structure, as we'll see here shortly. As far as loading these materials, the kind of model probe that we selected to start off with is a molecule that's called aerosol OT. I'll refer to it as AOT. And the reason why we chose it is it's one of few well understood surfactants that form reverse micelles in aliphatic solvent. And so we really wanted to form some larger structures versus dispersed molecules in these gels to track diffusion of. And based on literature, we expect that we'll get small reverse micelles in our aliphatic mineral oil solvent. For proof of that, we did small angle X-ray scattering, so I'm showing you a couple of curves here on the left, which I'll get a lot more into on the next slide. But the general takeaway here is we can formulate gels that have the same amount of copolymer, in this case 28% of our triblock copolymer, which I should point out is about 1/3 polystyrene N blocks, and is 125,000 grams per mole total, MN, number average molecular weight. And we can combine that either with oil, our aliphatic mineral oil, in the absence of AOT, which is the black curve here, or containing 1 weight percent AOT, which is the blue curve. And the basic thing you'll see here is that a lot of the features are very similar, especially at lower Q, so left half of the graph here. But as we get to higher Q, all the features kind of lift up, and especially at 10 to the -1 inverse angstroms forward, we see a raised plateau compared to the case with no AOT. And it turns out that this specific plateau indicates the presence of reverse micelles. And so we can vary the amount of AOT to get a better understanding of what's going on in these systems. Again, fixing the triblock copolymer concentration and varying the amount of AOT as indicated on the bottom left here. Again, a lot of these features that arise from the block copolymer, which I'll come back to, remain basically the same. And we see this plateau that I mentioned before lifting more and more with AOT concentration out to the left. And so we can model these things, and basically we model them as a set of two kind of bidispersed spheres, one that owes to the polystyrene N block domains and one that corresponds to the AOT reverse micelles. When we do that, we get really nice fits for the most part. And we see, just like I mentioned before, this model captures this plateau lifting with AOT concentration in these gels. And what the model basically tells us is that, first off, the size of the reverse micelles remains constant across changes in AOT concentration, and the core specifically is about 1 nanometer in radius. And also that the volume fraction of reverse micelles in these systems is linear with the weight percent loading of AOT. And this tells us basically that the AOT that's added continually forms more reverse micelles. And we have separate experiments, which I'm not going to show data for here, that confirm that we're above CMC at basically all of these concentrations and over all of the diffusion experiments that I'll show you data for later. And so we've got a lot of AOT reverse micelles and very few single AOT molecules in these systems. Now, when I get to talking about diffusion, one of the parameters that will be important that we don't actually measure here, but we take from literature, is the hydrodynamic radius of these reverse micelles, which has been determined in a couple of different publications to be right around 1.7 nanometers. And so now we kind of have a fuller picture of the structure of these things in our gels. Other than that, there's not a lot of exciting things happen here. We're basically adding more AOT reverse micelles as we increase the AOT concentration. I will also mention before I go on, there is a sharp diffraction peak here at about 3 times 10 to the -1 inverse angstroms. And this is actually from crystalline AOT. This is a specific diffraction peak for that phase. And we see it cropping up right at around 1 weight percent AOT. And so all of our experiments moving forward use exactly 1 weight percent AOT because we want to maximize the amount of AOT in our gels while avoiding having any crystalline AOT present. All right, so then the more exciting piece to this is we can vary the triblock copolymer concentration. And this is still the same 125,000 gram per mole, 33% styrene triblock copolymer. In this case, we fix the amount of AOT in these systems. And so from bottom to top here, we're looking at 5 weight percent SEBs at 5 increments of 5 up to 40 weight percent SEBs at the top. We can use the same model. And now I'll pause and take a second to talk through some of the features we're looking at here in terms of the copolymer assembly behavior. First, there's a sharp peak kind of at lower Q, relatively speaking, which corresponds to the separation of the polystyrene domains of the physical crosslinks in our systems. And then second, there's a series of broader features kind of in mid-Q range here, again, relatively speaking, that correspond to the size of those crosslink domains. And so as you can see here qualitatively, the sharp peak at low Q moves to the right in terms of Q space. And so we're seeing a decreased spacing between polystyrene domains. The broad features in the center are moving to the left. And so we're seeing polystyrene domains that are getting larger with concentration. And if we model these things, again, we see information about the AOT reverse micelles, but we also get these two geometric features quantitatively that I just mentioned. And we can plot those results and see exactly what I just mentioned. The radius of the polystyrene domains increases with triblock concentration. The spacing between them, kind of the distance between surfaces of these crosslinks, decreases with copolymer concentration. And also what I'm showing here in open and closed symbols are the cases where AOT is present and not present. And so we basically see really good agreement, just like I talked about on the last slide, between 1 weight percent being in the gels and 0 weight percent being in the gels. So it really makes no difference there. Now, as we start to move towards thinking about the transport properties of these materials, this is all really informative, but there's one other parameter we need to consider, which is kind of overlooked in the sketches that I've been showing you so far. So if I zoom in to one of these green regions where in actuality there's a lot more mid-block chains, the mid-block chain density is much higher, there's what's called the mesh size, which is a very common term among covalently cross-linked gel folks and hydrogel folks. And the mesh size describes the distance between chains in this green space. I should have mentioned before, while the purple regions are glassy crosslinks, all these green regions are fluid and very rubbery. And so there's a lot of thermal motion here where the diffusion will occur. And so this mesh size is likely to also be very important in describing the diffusion of our reverse micelles through these systems. In terms of the mesh size, it's very difficult to measure in these systems because of the electron contrast between the other phases, but we can do a calculation based on some scaling theory originally postulated by Pierre-Dijon, which defines the mesh size as a combination of the radius of gyration, in our case of the mid-block chains, the overlap concentration of these chains, and the actual concentration of the mid-blocks within the green space. So this is an overall concentration, but the concentration of ethylene butylene chains within the green matrix. And that calculation results in this trend here. Basically, again, as we increase the triblock copolymer concentration, the primary factor being changed here is this concentration in the numerator, and we see the mesh size decrease over that range. And so kind of to summarize this overview of the structural behavior of these materials, it's important to realize that the mesh size is on a much smaller scale than the other dimensions that we were talking about, as shown by the dark green line at the bottom here. And so that'll come back into play when we start talking about transport properties. Okay, so we built up this nice structural picture, but like I said before, we really want to connect structure with formulation and transport properties. And so the next thing we need to do is try to get a handle on what are the transport properties of our AOT reverse micelles through our thermoplastic elastomer gels. And so to do this, we take kind of a couple of pieces from other areas. One is from release or drug diffusion in hydrogels, which uses this kind of release experiment where we take a gel loaded with AOT, we drop it in pure solvent. In this case, we match our gel. So this is aliphatic mineral oil. And then over time, the concentration gradient drives the AOT reverse micelles from the gel to the surrounding liquid. And then to analyze the remaining concentration of AOT in these gels, we actually use FTIR spectroscopy in transmission mode through the gel. And there's a very well-resolved carbonyl peak that corresponds to groups in AOT and none of the groups in the polymer. And so we can track that peak absorbance over time. Here, this is from black to light green, indicating the progression in time. And as you can see, the height goes down because the AOT is releasing from the gels. And therefore, we're seeing this diffusion that I was just talking about. This is some example data for a triblock copolymer concentration of 20 weight percent initially, and an AOT concentration of 1 weight percent initially. And then we can take these and basically plot up a release curve with time. In our case, because we're looking at things in terms of the gel, whereas others have traditionally looked at the surrounding solution, we're looking at what we refer to as the retained mass of AOT in the gel. And so initially you have 100% AOT in the gel, and you see it going down over time as we approach roughly 0 as all of the AOT is being released. So we can do this for all of these different gels that I showed you structural information for, from 5 to 40 weight percent triblock copolymer. These all initially have 1 weight percent AOT. And as you might expect, the more copolymer concentration there is, the slower the release of AOT from these gels. We can kind of quantify that, and there's some complication, which I'll come back to in a second. But we can quantify these behaviors using a first order release model shown here. The primary thing I'll point your attention to is the first order release constant K, which describes the rate of release from these gels. And we see, as we observe on the left here, that the K value decreases as polymer concentration increases, suggesting that the release of AOT is slower with increasing polymer concentration. Now, before I go on, I guess I should point out the kind of challenge with these systems is that if we take a thermoplastic elastomer gel and submerge it in the mineral oil that it's also composed of, these materials swell considerably. In a lot of cases, outside of the lower compositions, 5, 10, 15 weight percent swell a little bit less, but 20 on up are nearly doubling in mass in swelling over this period of a few hundred hours. And so it's very difficult to model something like diffusivity that would be nice to have in order to understand theoretical connections between structure and diffusion of AOT reverse micelles. And so the next thing we did to move forward and get diffusivity, instead of addressing it theoretically, we went back to the experimental planning board, and we incorporated a new step before our diffusion experiments start. So basically what we do now is we incorporate pre-swelling, pre-diffusion experiment swelling into our trials, wherein we take the same gel as before, we submerge it in a surfactant, an AOT surfactant solution of mineral oil at the same exact concentration as the gel. And then once the gel is submerged in this oil with AOT, there's no concentration gradient. So there's no desire for the AOT to elute from the gel. And so this isolates the swelling step to this first half of the process. And then once the gel has reached its kind of an equilibrium swollen mass, we remove it from this pre-swelling solution and then repeat the experiment that I talked through before, wherein it's submerged in pure mineral oil and there's a concentration gradient which derives the release of AOT from these gels. And so there's a couple of things to be wary of here, just be aware of, which is as these gels swell, their structure changes. We're adding more mineral oil into the system, and so the triblock copolymer concentration is naturally going down. As you see on the x-axis here, these are for 5, 10, 15, all the way up to 40 weight percent initially triblock copolymer gels. You can see their swollen compositions are considerably lower because of that doubling of the gel mass through incorporation of mineral oil. But in general, the polystyrene domain size doesn't change. Not much of that mineral oil, if any, is going into the polystyrene domains because it's chemically incompatible. The x-axis values won't match up here, but just for comparison to what we started at, these lighter points are the gel structural parameters before pre-swelling, and the open symbols are post-swelling. And so like I mentioned, the radius of the polystyrene domain is staying mostly constant, whereas we see a serious dampening in the drop of spacing between polystyrene domains based on the swelling behavior. So just something important to keep in mind. The other thing here is that the mesh size calculation doesn't change based on triblock copolymer concentration. The biggest thing to adjust for is the fact that instead of 40 weight percent of our highest concentration now, our gels start around 19%. And so we're just examining a different part of the curve in terms of our structure. So now, like I said, we repeat the same transport experiments. Not a lot experimentally changes except for we're now handling pre-swollen gels. And so the triblock copolymer concentration is quite a bit lower, but we still get very similar release curves, and they still correspond to faster release for lower triblock concentration and slower release for higher triblock concentration, except for now we can model them using fixed second law with appropriate boundary and initial conditions. And instead of those first order rate constants, we extract diffusivities of AOT reverse micelles through the gels, as indicated by the dark green points here. And so just like before, except for I think the experimental parameter is a little bit less empirical. since we have diffusivity as a function of triblock copolymer concentration in the gels, we can model it using one of various different solute diffusion models through gels. In this case, this is a hydrodynamic model for a homogeneous gel, which we feel our materials fit the bill well for that type of model. And you can see the fit is pretty good. It's important to note here, this model, the scaling of this model is derived from mesh size arguments. And so this isn't taking any of the nanostructure of the copolymer into account beyond the mesh size calculations I showed you before. I should define here that there's a typo here, sorry. This should be D on the left-hand side is equal to D0, which is diffusion through pure solvent, in other words, zero polymer concentration. Kc is a constant that corresponds to the polymer solvent system being examined. RH is the hydrodynamic radius of our probe, so that's the 1.7 nanometers I mentioned before. And phi EB is the volume fraction of ethylene butylene chains in the green space of the network. So this is interesting, maybe not totally unexpected, and I think an analogy is really helpful here because we have a lot of structural things going on. And so if you imagine reverse AOT reverse micelle being a person in pre-COVID times working their way through New York City, you might imagine that the buildings are like the polystyrene crosslink domains, and the streets are the continuous space where the AOT can diffuse. But other polymer chains are like other people. So the mesh size is being described by the other people in the street. And so in this analogy, you can imagine that the other people are going to be a lot more restrictive to this person, this reverse AOT micelle, moving through Times Square, for example, than the surrounding buildings are. And so maybe it's not such a bad finding, or it's not so surprising that mesh size is the primary player in diffusion through these materials. The final kind of test we did to explore this further was to use a few different triblock copolymers varying in molecular weight. These all have approximately the same fraction of polystyrene at 33%. But because of their different molecular weights, they have different polystyrene radii, as shown here. They have considerably different spacing between the polystyrene domains, from 26 for the lowest molecular weight up to 46 for the highest molecular weight, but their mesh size is approximately the same. And so this kind of gives us a tailoring of one parameter in pretty vast change, and while fixing the other one. These are all the same polymer concentration. This is 10 weight % in the swollen state. And what we see is that the diffusivities are very similar for these three different gels. And so this further strengthens our belief that the mesh size is the primary controller in terms of diffusivity through these gels. We haven't had time to go through the small angle X-ray scattering analysis in full form yet, but we do have transport experiments for 5 to 40 weight % for all three of these copolymer molecular weights. The 70,000 gram per mole series is in red. The one we were looking at before, the 125,000 gram per mole series, is in green, and the 170,000 gram per mole series is in blue. And basically what we observe here is that the same hydrodynamic model with the same value of Kc and D0 fits the data very well. And so this further establishes, we think, that the mesh size is the main driver in dictating diffusion through these systems. And we're finalizing our X-ray scattering, and we hope that this will further kind of clarify this idea. So I know I just have a couple of minutes left. That's the end of my